Hey, it's me, Bruno. Welcome back to Bruno's Biologs, where we break down what's new, what's next, and what's weird about spatial biology. Today's episode is... Wait, what was I talking about again? Oh right, memory. Because whether it's forgetting your keys or reliving the moment you aced that thesis defense, memory is one of the most powerful and sometimes puzzling parts of being human. And spatial biology is giving us new ways to see how memories are formed, stored, and maybe even lost, all by keeping cells exactly where they are. So what's going on in the brain when a memory forms? Researchers recently set up a clever experiment to tag the brain cells active during memory recall. Using a genetic labeling technique, they marked these memory-associated cells with a red fluorescent tag. Behaviorally, the mice froze more during recall, confirming that memory had truly formed. Then, with spatial transcriptomics, they mapped where those memory tag cells were sitting inside those basolateral amygdala. You can see here, the memory engram cells aren't random. They organize into real functional networks. Zooming in, they found that excitatory neurons involved in memory showed distinct changes in gene activity compared to non-memory cells, turning on specific programs that are likely to help stabilize long-term memory. But neurons weren't the only ones involved. Astrocytes, those star-shaped support cells, were activated too. Astrocytes surrounding the active neurons ramped up their own gene programs, showing that memory formation is a coordinated multicellular event, not just neurons doing all the work. And memory isn't just the only thing spatial biology is uncovering. In another study, researchers used xenium spatial transcriptomics to map how cells in the brain's ventricular-subventricular zone, the birthplace of new neurons, talk to each other during brain repair. These spatial maps show exactly where key receptors like LGF1R and LIFR are expressed across neural stem cells and supporting cells. By seeing where these signaling molecules are in the tissue, not just that they exist, we can start to map out how regeneration might actually happen. Look, I know this kind of standardization might sound dry, but it's really the foundation of why we need to make spatial data truly scalable, comparable, and usable in both discovery and translational science. So whether you're trying to understand the biology of memory or just want to make sure your data holds up across the field, spatial biology has changed the way we study the brain. Thanks for watching. If you're working with the brain tissue or thinking about just spatial tools or just want to geek out over data, hit me up, let's connect. Until next time, keep asking big questions. I'll see you in the next Biologs, and thanks for watching.